So hi, I'm Tori Wiedorn from Boston University in Thrust Area 2, and today I'm going to be introducing Jonathan Kimmelman, PhD, who is a bioethicist, media scientist, and associate professor at McGill University in Montreal, Quebec, Canada, who studies drug development and risk. He has been published in numerous media outlets, including the New York Times, Globe and Mail, and BBC World Service. He also has served in an advisory capacity for the World Medical Association, FDA, and the Canadian Institute of Health Research. Kimmelman formerly chaired the Ethics and Public Policy Committee for the International Society of Stem Cell Research, where he led revisions of the Society's ethical guidelines for stem cell research and clinical translation. He has served as a member of on 3 National Academy of Medicine committees, including one advising NSA on health standards for long duration and exploration space flights. NASA.
I do not advise NSA.
Sorry, sometimes
okay.
He is a deputy editor at Clinical Trials Journal and an associate editor at PLOS Biology. Kimmelman currently leads a group of researchers in a collaborative effort called STREAM. STREAM, or Studies of Translation, Ethics, and Medicine, is a research collaboration centered at McGill University. The network includes affiliates across North America with shared principles about the goals and methods for studying clinical translation. Members work in several medical specialties, such as ethics, epidemiology, biology, psychology, among others. According to the STREAM website, translationalethics.com, their mission is to apply empirical and physiological tools for addressing scientific, ethical, and policy challenges in the development and translation of health technologies. The primary goal is to furnish scientists, ethicists, policymakers, and others with high-quality evidence and analysis concerning clinical translation. So, thank you, and here he is.
So, what I want to do is go over 2 basic sets of issues. So I take it that this program is aimed at helping you to kind of figure out how to develop products that can be used in human beings. And I want to cover 2 sort of ethical dimensions about trying to develop products for human beings, medicines, devices, etc. The first I'm going to cover in the first part is about the ethics of human experimentation and drug trials. And in the second part, I want to talk about some challenges with reproducing laboratory research, particularly as we try to move laboratory research into clinical applications. Okay, so let me start with the first part. So drugs, devices, vaccines are just really awesome things. They do great things for us. If you add up, for example, the number of human life years that have been saved by highly active antiretroviral treatment since its development, 2.8 million years of extra life. If you look at pediatric malignancies back in the 1950s, survival rates, if you had cancer as a child, were about 10%. Today, they're upwards around 80%. So drugs are great things. But the problem is that they have a downside. Number one, they're incredibly expensive. It's one of the most rapidly growing healthcare expenditures. This is a little bit dated, but pharmaceutical prescriptions, for example, account for $234 billion of health expenditure. So we spend a lot of money on drugs, devices, vaccines, et cetera. And obviously that eats up a very large fraction of our healthcare budgets in the US, something about in the order of 17% of our healthcare, of our entire GDP is allocated towards healthcare. That every dollar we spend on healthcare is a dollar we don't have available to spend on things like education, environment, foreign aid, et cetera. So drugs, devices, vaccines, the things we're trying to develop here are expensive, and they're also really dangerous. If you look at mortality due to adverse drug reactions, it's between the 4th and 6th leading cause of death. Just taking drugs in the wrong way can be lethal. So what this means is that if we really want to unpack the value and potential and impact of drugs, vaccines, devices, et cetera, while not having to confront some of the toxicities, or making sure that we're getting good value for these, we really need high quality evidence that they're useful and that they're safe. And herein lies the challenge. In order to acquire that evidence, you have to do experiments. not just any experiments, but you have to experiment on human beings. And that is ethically fraught. And it's ethically fraught for a couple different reasons. The first reason why it's ethically fraught is because your reagents, your research reagents, have what ethicists call moral status. So think about this. Everyone here probably has worked in a lab. When you go to the lab, you go to grab tris buffer off the shelf or, whatever. When you start making that, your solution, you start making your reagents, those solutions never talk back to you. They, if you're a geologist and you start breaking open a rock because you want to characterize its chemistry, et cetera, the rock never says, "Don't break me open, that hurts. Please don't do this, I have other plans." Whereas human beings, if you want to study them, if you want to open them up, you are potentially inflicting pain on them, and you can be frustrating their goals. And so the very research reagents that you use to collect evidence, to use drugs in a way that's safe and effective, requires that you experiment on entities that have moral status, that have the capacity to suffer as a result of your doing research on them. Further complicating the ethics of doing experiments on human beings is that the very people who are licensed to do those experiments are doctors. Now, doctors, of course, swear by the Hippocratic Oath to first do no harm. And so here you have a doctor who is, on the one hand, sworn to advance the best interest of their patient, while on the other hand, sworn to advance the best interest of science. And so the duties of a physician researcher are, in some sense, in conflict. So that makes conducting human research very sort of ethically complicated. And much of what I'm going to talk about in the first section of this presentation is about how we have erected a sphere of protection around the human subject, around the patient, when researchers conduct research on human beings. In the second part of the lecture, I'm going to talk about another set of issues that are ethically fraught when we do research on human beings, or even research on non-human animals. And that's that the evidence that we produce when we do research on human beings is intended for human consumption. Much in the same way that if you are a farmer and you're growing food that's going to be eaten, you want to make sure that your food is not adulterated through the processing. In the same way, you want to make sure the evidence that you collect when you experiment on human beings is going to be clinical grade. It's going to be of the quality that physicians can use at the bedside to decide what kind of care to give a patient, and to make sure that the risks that patient's going to endure are going to be outweighed by the prospect of benefit. And so in the second part of the talk, I'm going to talk about the various ways that we have tried, I think unsuccessfully, but we've made some progress, in trying to protect the downstream users of evidence by making sure that we can maintain the integrity of the science that we produce. So in the first part, I'm going to go through some really quick history of medical research to put everything in context. Then I want to talk a little bit about the history of medical research ethics. And then I want to give you an overview of what are sort of the core principles of human research ethics. So let's start with the history of human research. In a sense, experiment on human beings is as old as medicine. There are a lot of really interesting kind of pre-modern examples. This is an example of Edward Jenner in 1796 conducting one of the first experiments on James Phipps of a smallpox vaccine. Jenner was a country doctor. He had postulated that exposing people to the pus from blisters from milkmaids who had milked cows with cowpox would protect them, would give them immunity against human smallpox. And so one of the very first sort of examples of a kind of prototypical human experiment was when he took a kid in his village, injected him with this pus, and then deliberately challenged him. He actually exposed him to human smallpox blankets that had been used by a smallpox patient. And that was really one of the first demonstrations of what later came to be a smallpox vaccine. Edward Jenner was very famous for that. There are a lot of really amazing and grotesque examples of human experimentation prior to the 20th century. Some of them are kind of funny, some of them are really odd and quirky. I always like to give the example of John Hunter, who was the physician to King George III. Since I'm down here in the United States, everyone here knows who King George III is. In Canada, no one's ever heard of him. But at any rate, John Hunter was quite a character. Prior to the 20th century, a lot of human research was actually self-experimentation, and he was very much in that tradition. he was actually trying to find the cause of gonorrhea, and he had postulated that gonorrhea, rightly so, was infectious. And so he did an experiment on himself where he took the pus of a lesion from one of his patient's penis and injected his own penis. Ended up getting gonorrhea, but he got 2 for the price of 1. He actually also developed syphilis because it turned out that that person also had syphilis. We're starting to move now into the more contemporary area, and this is sort of the cusp between what used to be kind of what I'd say is pre-modern human experimentation and modern human experimentation. This is Walter Reed. So Walter Reed was a physician in the U.S. Army. His physicians exposed themselves to mosquitoes and to see whether or not they triggered yellow fever. And in fact, one of his physicians, Jesse Lazier, who was a very well-trained and young, promising medical scientist, ended up developing yellow fever and dying. Very expensive way to do medical research. You train someone in science, in medicine, they apprentice with you, and then they die in their own self-experiments. So there's got to be a better way. And the solution that they came up with was hiring Spanish workers to expose themselves to yellow fever, giving them, paying them something like $100. They get extra benefits if they die. There was funeral costs that were covered, et cetera. It's dark. The history of medical research is pretty dark. But the point is that here you have this transition point. This is, again, in the early 1900s, late 1800s, where experimentation is shifting mainly from self-experimentation to experimenting on others. Now, for the most part, medical research was kind of a cottage industry. It was not really a large-scale operation, and that changed around World War II. And there were a couple of different factors that changed it. Just 2 I'll mention really quickly. The first is World War II. So prior to World War II, there really wasn't all that much organized government-funded research anywhere in the world. And part of what was perceived, rightly or wrongly, as having been integral to the Allied success in World War II was the fact that the US government had lavishly funded lots of engineering and scientific research, including research that led to the development of the atomic bomb. After World War II, that model of the government giving money to researchers, no strings attached, continued in the 60s, and that's why we have the growth of the NIH as a major funding agency. And what that did is essentially create resources, supply resources for doing much more biomedical research, and that really sort of slingshots the US into the forefront of biomedical research in the world. So that's one of the forces that drives a growth from cottage industry human research into kind of industrial scale. The other big factor is the rise of drug regulation, the Food and Drug Administration. So, back before the 1930s, for all intents and purposes, there really wasn't any kind of regulation of drugs. If you wanted to market a drug, all you had to do prior to the 1930s is make sure that you labeled accurately the contents of your drug. So if you were selling snake oil, all you had to do is make sure you explained that the oil was, accurately derived from a particular snake. You could make whatever claims you wanted about efficacy and safety. You just had to make sure that you were accurate about what snake it was from. And that changed in the 1930s, around 1936. The history of drug regulation, I'm just going to say as a footnote, and the history of medical research ethics is basically a history of scandal, and debacle followed by policy response. And in particular, anytime you see a really big policy change in bioethics, look for a trail of dead children, because it's usually dead children that triggered policy responses. Hasn't quite made it there with gun control yet, but but at least in drug regulation, it tipped us over. And so the first really big scandal that drove the development of drug regulation was the sulfonilamide elixir scandal, which was this was a pre-antibiotics. This was sulfa drugs were the mainstay for treating infection. Very bitter, hard to get kids to take these drugs. So a company called Massengill Company down in Tennessee figured out a really ingenious solution, which was they dissolved sulfa drugs in polyethylene glycol, which is very sweet apparently, and it was easy to get kids to take the sulfa drugs that way. Probably everyone here knows, or at least some of you probably know, that is not a good idea. It turns out that polyethylene glycol, also known as antifreeze, is toxic. And so over 100 kids ended up dying from that. Congress then empowers FDA to require that there be demonstration of safety before drugs actually get marketing approval. And so we're halfway there. FDA in the 30s and 40s now requires demonstration of safety, but not demonstration of efficacy, and that changes with the next big scandal or, problematic episode, which was the outbreak of phocomelia, or limb stunting, among children whose mothers had taken thalidomide. Thalidomide was a drug that was being used in Europe for management of morning sickness and anxiety, and there were a couple cases that have been reported in the medical literature of children being born with stunted limbs. It had not yet been approved in the United States. A very young, they applied, the company that made the product, Grunenthal, had applied to the FDA to get that product licensed. And there was a very young pharmacologist, a woman who was a McGill grad, I'm proud to say, Frances Kelsey, who had read the reports in Europe about the outbreak of phocomelia and had been pounding on the door of the commissioner saying, do not approve this drug in the United States. And when it became really clear that thalidomide was causally linked to this, the FDA did not ultimately license the drug, and it was that image of that near catastrophe in the United States that drove a series of amendments to FDA policy that for the first time required that you demonstrate not just safety but also efficacy to market drugs and then later devices, vaccines, et cetera. So think about what this does. If you're a drug company and you want to market a product, you have to go through regulatory approval, and that regulatory approval requires that you demonstrate safety and efficacy. And so now you have this incredibly strong financial incentive to be doing high-quality human experiments. And that, probably more than any other factor, drives human research from kind of an interesting, kind of quirky thing that physiology professors are doing, and research, and hospitalists are doing, to something where there's actually a lot of money and industrial scale efforts in trying to generate evidence in human beings. Okay, so let's switch to talking a little about the history of medical research ethics. So medical research ethics grows more or less in parallel with the emergence of human research. There are pre-modern statements that sort of get at the fact that lots of prominent medical scientists were thinking a little bit about the ethics, but there was nothing really formalized. There were no sort of clear policies, certainly no regulations. So Claude Bernard, a very famous French physiologist, kind of the father of physiology, once said in one of his writings that one should never perform on man an experiment which might be harmful to him to any extent, even though the result may be highly advantageous to science. A very apt intuition, but not one that necessarily was shared by most other physicians who were doing research on human beings. Around the turn of the century, the same time that Walter Reed is doing his experiments down in Cuba on yellow fever, a lot of the thinking about human research and the ethics of human research is really tied up with the way that people think about vivisection. So today we think of vivisection or animal experimentation as a completely different set of issues than human experimentation, although they are actually coming together. If you're working at the cutting edge of bioethics, these two areas are starting to re-merge. But until recently, we thought of these as kind of fairly separate entities. But a lot of the movement to try to formalize the protection of human subjects comes out of concerns about anti-vivisection. And the idea is that if they're going to be experimenting on stray cats and dogs, the next step is going to be experimenting on orphans, human orphans. And if they start experimenting on orphans, the next thing that's going to happen is they're going to start grabbing middle-class children and starting to experiment on them. So we got to draw the line somewhere. We're going to stop at animals. So some of the first sort of thinking about medical research in terms of policy and regulation is beginning to emerge at the turn of the century around animal research. But as I said earlier, what drives this kind of inchoate thinking about ethics into formalized policy are a series of scandals. And the biggest scandal of all, the founding trauma that leads to bioethics, is of course the Nazi doctor trials. These were trials that were organized by the Allies, in particular by the United States, to prosecute Nazi physicians who had conducted utterly grotesque experiments on concentration camp inmates as part of the Nazi war effort. I can give you lots of examples. This is a picture from high altitude experiments that the Nazis were doing to look at the limits of human survival with fluctuations in air pressure. There were lots of experiments looking at prolonged immersion in sub-freezing temperature water to see how long human beings could survive. This was relevant for the Nazis. Their pilots were getting shot down in the North Sea. And the question Nazis always had, any military would have, is should we send a rescue operation or not? And so knowing something about how long humans could survive in freezing Arctic temperature water was something that the Nazis would have wanted to know. Lots of other experiments, I mean, just really horrible stuff that you probably don't even want to hear about. But nevertheless, after World War II, the Allies orchestrated a series of trials, of which the Nazi doctor trials is one of the major ones. And there are 23 Nazis that are condemned, that are found guilty of human rights violations in that. And that leads to the very first formalized articulation of what you can and can't do when you experiment on human beings. Up until then, people had kind of resisted formalizing this. The idea was that doctors are professionals, we can kind of trust them to exercise their judgment. And this really begins to change things, where we start saying, you know, maybe we shouldn't be relying on just doctor discretion when we think about what they can and can't do with human beings. So the very first policy that's articulated is called the Nuremberg Code, after the city where the Nazi trials were held, Nuremberg, Germany. And the Nuremberg Code articulates 10 different principles. I'm not going to go through these, we can get to these later, but the one that really we need to pay attention to the most is the very first principle. And it's provision 1, and I'm going to read this to you, and I'm going to ask you to tell me what's wrong with it. So it says, the voluntary consent of the human subject is absolutely essential. This means that the person involved should have legal capacity to give consent, should be so situated as to be able to exercise free power of choice, without the intervention of any element of force, fraud, deceit, duress, overreaching, or other ulterior form of constraint or coercion, and should have sufficient knowledge and comprehension of the elements of the subject matter involved as to enable him to make an understanding and enlightened decision. So this is the very first articulation formally of the requirement of informed consent, as the Declaration of Helsinki, articulated in 1962 by the World Medical Association, which is kind of like a UN of different national medical associations. So the American Medical Association, the Canadian Medical Association, British, et cetera. They come together, they articulate the Declaration of Helsinki, which basically recapitulates Nuremberg, but they carve out a space, and I'll get into it later, about how you can do research in patients who lack legal capacity or competence to give informed consent. Now, the second big trauma that I think drove more formal regulation of human research was the so-called Beecher expose. So this was basically an article that was published in New England Journal of Medicine by this guy, Henry Beecher, who's a very interesting and kind of strange character, an anesthesiologist at Harvard. If we have time, I can talk about why exactly he was strange. But what he did is essentially he went to top medical journals, browsed through them, and found 22 examples of research that were flagrantly unethical, that had been published in top-tier, U.S. medical journals, performed by credible medical scientists. And his point was that we had created this great policy with Nuremberg that required informed consent, that required that we balance risk and benefit, et cetera, and that clearly the U.S. was not honoring the spirit of Nuremberg. And this really sort of drove home that there was clearly a double standard in terms of how U.S. and other researchers, British researchers, saw their research compared with, say, Nazis or physicians that were in discredited regimes. Now, the key insight in that expose was the following. I kind of alluded to this earlier. Normally, if you're a physician, you owe your duty solely to the patient. Your job is to do the best you can for a patient. When you are a physician investigator, you are legitimately dividing your loyalty between the patient and their welfare. You're still a doctor, you can't abandon your Hippocratic oath. And you're also committed to society, because after all, you're generating evidence that's aimed at trying to improve the health care of future patients. So Henry Beecher's solution to this was that we could rely on physician discretion, good physician professionals, they get good training, they're going to know how to navigate that conflict of interest. That view has been discredited, but the basic insight that physician investigators endured a conflict of interest or divided loyalty when they do research is one that we hold still today. So we have the Beecher expose, and then in 1972, revelations about the Tuskegee syphilis study. How many people here have heard of Tuskegee syphilis? Okay, good. This is great. Canadian audiences, not so much. So this was a study that was an observational study looking at the progression of middle to late stage syphilis in African American men in a rural part of Alabama. Just to say really quickly, these men were withheld from what was standard of care for management of their syphilis. They were not really given proper informed consent, lots of blood draws, spinal taps, et cetera. And the person of the public health service, all this had been done in in the light of day. The studies had been started in 1934. They'd been published in a series of articles, major public health journals. There was no secrecy about this at all. No one had sort of blown the whistle until a guy named Peter Buxton at the Public Health Service read about this and said, this is nuts, this is crazy. So he blew the whistle on it. The Public Health Service organizes an investigation. They ally with the American Medical Association, and they come to the conclusion, nope, there's nothing problematic here at all. This is all ethical stuff, no problem. Not the most enlightened organization, AMA, at least historically. Peter Buxton wasn't happy with that. He took all the materials, sent them over the transom to the Washington Star, which was the main newspaper in Washington, D.C. at that time. And the next day, there are headlines about what's going on in these studies. And Congress, then the U.S. Congress, organizes a series of hearings that lead to the very first formalized policies and regulations on human research. And there were a series of revelations during these congressional hearings of other experiments. This is a Willowbrook study. Willowbrook Hospital, this was a school for children who were severely developmentally delayed. A series of experiments there where children were deliberately infected with hepatitis A in order to develop a vaccine for hepatitis A. Lots of other experiments that were conducted on U.S. soil that today we would think of as being very problematic studies. Congress organizes these hearings and asks basically a group of philosophers and some theologians and physicians to write up a report that is going to articulate for the first time the ethical principles that ought to govern human research. And that comes to be known as the Belmont Report. Probably one of the last times in U.S. history where philosophers are asked to make policy. But anyway, so the Belmont Report, written in 1978, and it articulates three core principles that ought to govern human research. The first is respect for persons. So what do we mean by respect for persons? It means that if patients have the ability to give informed consent, that you require always that they give informed consent before you do anything, any kind of research on them. If they can't give informed consent, well, we have these other kinds of provisions, surrogate informed consent, et cetera, and we limit the amount of risk for children, patients with advanced dementia, et cetera. So the second principle they articulate is beneficence. Not to be confused with benevolence, it's beneficence. And that just simply means that we always want risks to be outweighed by benefits to the human subject, if any, plus benefits to society. So there's some research that has no benefit to human subjects. That can be okay as long as there is sufficient benefit to society from the research, although there are limits on that, and we'll get to that. So the key point, this gets back to the Beecher, is that in order to make that risk-benefit judgment, better not to leave that to the discretion of physician investigators. The Belmont Report says that this ought to be mediated by these independent arm's-length committees that in the US are called IRBs, Institutional Review Boards, and they basically are refereeing that divided loyalty that physicians have between patients and future patients to make sure there's a favorable risk-benefit balance. So that's the second principle. And the third principle is the principle of justice. Again, you look back at this is the 1970s. I'm not sure justice would quite make it in there if we were doing Belmont today, but nevertheless, it's there. And what the Belmont authors mean by that is fair subject selection. So what does this mean? Well, when we do research, we always are picking out populations. We're always saying, here's a population of patients with, HIV or with cancer, et cetera. And what's meant by justice is that there be concordance between the populations that we're studying and the populations we're intending to benefit. So another way to put that is, it's unethical to build medical progress for advantaged populations on the backs of disadvantaged populations. That's unfair. Why should disadvantaged populations endure risks for the sake of advantaged populations? So that becomes canonized into regulations in the United States, there's three things I want to point out. This might be a little bit abstract about these principles that people often don't understand, but it's important that they understand them. The first is that they're not meant to be ranked. So when I say respect for persons is the first principle, beneficence is the second one, there's nothing implied there about respect for persons being more important than beneficence. So just because justice is ranked is the third one doesn't mean it has less priority. There is a chronological relationship. If a study does not have a good risk-benefit balance, there's no point in getting informed consent. So some of these principles get invoked earlier in considering a study than others. And the third one is that they're not fungible. In other words, you can't buy off a deficit in one principle by having a surplus in another principle. You can't say, yeah, this study is like is a really like high risk study. It's not really that beneficial. It doesn't really have a good risk benefit balance. But no problem, we're going to get like really good informed consent. We're going to make sure everyone really understands how much risk this involves. It don't work that way. You have to have each one of these principles has to be independently fulfilled. So these principles now get embodied into regulations. In the US, the regulations are called the Common Rule, or 45 CFR 46 if you are legally inclined. FDA has its equivalent, 21 CFR 50, and there's a couple other policies. You don't need to know, you don't need to memorize those numbers. All you need to know is that there are regulations that say you got to get informed consent when you do research on human beings. And there are a lot of other policies that are out there. I just want to mention really quickly, there's a policy called CIOMS, that's the World Health Organization's policy, more or less recapitulates the same principles that's articulated in US regulations. And if you're in drug development, you'll probably hear a lot about this policy, the International Council of Harmonization Good Clinical Practice, which is a policy that is meant to set standards on demonstrating that your research was performed ethically when you submit INDs, applications for drug approval or device approval, whatever it is, to regulatory agencies. So FDA, the European Medical Association, Japanese, et cetera, they all subscribe to GCP, and that means that those trials all have to demonstrate they got informed consent, they had independent review, et cetera. So it embodies some similar ethical notions. Let me say a couple things really quickly to kind of unpack how we actually operationalize these principles. And I'm going to go through this really quickly, but it's just, it's good to kind of have a quick tour. So what do we mean by favorable balance of risks and benefits? Well, the way that we operationalize this largely is through taking an experiment, a human experiment, and dividing it into those components that are sort of therapeutic components. So if you're doing a drug trial, you're giving a patient a drug that may have therapeutic quality. But also you're often collecting blood samples, doing imaging, lots of other aspects of the study that have no therapeutic benefit at all. There's no therapeutic potential. It's purely done for science. And those are research procedures. So each one has to be disentangled and evaluated separately from each other. And the drug component would be evaluated using a principle of clinical equipoise. And all clinical equipoise is, it's kind of a lot of people have a hard time with this concept. All it means is that when you're studying a drug or a device or a vaccine in human beings, you can't deprive them of standard of care. You can't deprive them of treatment they would get outside the study. So if you're doing a randomized controlled trial, a study with a control arm, patients have to be randomized either to standard of care or a new treatment that is believed to be competitive with standard of care. It's not proven yet, but it's believed to be potentially better, but you don't know, it could be worse. That's all that clinical equipoise means. So that means if you have a new drug and there's a standard of care that you know works for those patients, it's unethical to use placebos. Okay? And then of course, research procedures, you want to always minimize the amount of risk. So if you can do with one, if instead of doing 2 biopsies, you can do 1 biopsy, the amount of patient burden and the value of the knowledge is a favorable one. Okay, informed consent. How do we get informed consent? There really are 3 core elements of what is embodied in informed consent. The first is capacity. You have to have a certain cognitive ability and emotional state in order to be able to give valid informed consent. There are various kinds of tests that you can give to patients to make sure they have the intellectual capacity to give informed consent. The second thing is that they need to have adequate understanding or comprehension. They need to understand what the risks, what the benefits are, what the alternatives are to participating in studies, et cetera. There are a whole bunch of different things that you want to make sure patients understand before you enroll them. sometimes people misconstrue the notion of informed consent. They say, there's no way a patient could ever understand, the research, why do we even bother with informed consent? That misunderstands what understanding is about. They just have to have adequate understanding. You don't have to do a PhD to go into a medical experiment. All you have to know is what is relevant for a patient to decide whether or not he or she wants to expose themselves to the risk. The higher the level of risk in a study, the more demanding you're going to be about understanding. So if you've got a really high-risk study, you might go through a discussion with a patient and you might actually quiz them to make sure they have adequate understanding before you enroll them. If it's low risk, you don't really have to worry too much. You get a sense they understand it. You don't have to quiz them necessarily. The most effective way to get valid informed consent is not through informed consent documents, which are these, very boring forms that people spend a lot of time reading. It's through face-to-face discussions, physicians explaining what the risks and benefits and alternatives are to patients. And the third component, so I mentioned capacity comprehension, the third component is voluntariness. What this means is freedom from coercion. A doctor can't say, I'm running a trial, I want to enroll you, and you should know if you don't go into the study, I'm never going to treat you again. You're not going to be my friend. That's coercion. That you can't do that, generally speaking, in medical research. Manipulation would be also a threat to voluntariness. What's manipulation? Manipulation would be withholding certain bits of information or shaping the information that you give patients in a way that's going to get them to enroll. So you say, hey, we're doing a study, and it's a pretty really interesting drug, and there's not really going to be that much burden. Are you interested? The patient says, yeah, it sounds really good. It sounds interesting. I'm willing to do it. And say, great. We're going to get the informed consent document. Oh, by the way, I forgot to mention, there's also a biopsy in this procedure. 2 biopsies. Are you up for that still? That's called lowballing. The patient's already made an emotional investment in enrolling in the study, and then all of a sudden the higher risk stuff is rolled out. That's a form of manipulation that sales tactic, car salesmen and women use, and something that we don't want to encourage very much in medical research. Okay, fair subject selection, I already talked about that with respect to making sure that we have concordance between enrolled populations and the populations that we're trying to benefit. I won't go into this too much. And the issue about value, which I'm going to talk about in a minute, means that the studies have to be designed in a way that's going to support valid inferences about clinical utility of a drug. Studies have to be addressing something of relevance. That it shouldn't be frivolous or medically irrelevant in terms of future health care. And when you do a study, people always miss this. It's important to remember, there's no scientific value if you do an experiment on human beings and you never publish the results. No one can learn from that. There's no way you can accrue scientific value. That's private knowledge, it's not public knowledge. The whole point of science is public knowledge. So reporting and publication is a really key component of protecting human volunteers. The fifth component here is independent review. I mentioned that you have these independent committees called IRBs that are refereeing the risk-benefit judgment and the informed consent process before you enroll patients. So it's important to remember that's prospective. You can't retrospectively go back and evaluate a protocol. They have to be arm's length or independent of the researchers. A bit of a joke, in my opinion, because we don't really have proper independent ethics review in any jurisdiction that I can think of. But anyway, it's supposed to be independent. I always just think it's strange when I sit on an IRB and I'm reviewing protocols from my peers who are in other departments who could affect my promotions. It doesn't sound too independent to me, but anyway. And then, of course, the people on the IRBs need to have requisite expertise and competence in the science to be able to, and the ethics to be able to evaluate those studies. Great. Okay, so far I've talked all about how we protect the reagents, namely the human subject, in research. I want to just close this segment of the talk by emphasizing that when you do research, you can think of it as having lots of different stages. You plan out your research in human beings, you've got your clinical trial, you get the results, you report it, and then hopefully people read those reports, they take them up, and they incorporate them into either products or into new studies. So far, most of what I've talked about is the protections and oversight during the stage of the trial. Right? Incipient to the trial, because IRB review is prospective, and then during the trial, you have these oftentimes committees called data safety monitoring boards that make sure that the risk-benefit balance and informed consent is still valid. So if you ever pick up the newspaper and you read about a clinical trial that was stopped early because patients were benefiting much more than expected, or it was stopped early because, patients, in the placebo group were doing better than the treatment group, that's because there's a committee called a Data Safety Monitoring Board that saw a trend in the trial, and they pulled the plug. And so so far, I've talked about sort of what happens, among these, in this time period among these two different entities. But as I said, there's a lot that's going on before that period and after that period, and there isn't a lot of really good policy and ethics that's going on in these other zones, and that's what I'm going to talk about in the second component. Before I go on to that, I want to just pause for questions, and I also, how much more time do I have? Where am I? Okay, I'll go through quickly the second part, but let me pause for questions because I want to make sure to reinforce this component here. So, yeah.
I guess for me it's a lot of... Who is in charge, I guess, of quantifying these factors? So, for instance, determining what a placebo effect, treatment might be, for instance. Like, if we have a disease that has no cure and the control group is, randomly selected, there might be, infants whose lives are at risk there, for instance. So I'm just wondering, like, you have all these protocols in place, but of course every study is different, and so the IRB, it's like their job to do that. And I'm just wondering, is that process usually, are there conflicting views sometimes? Is it very hard for people to agree on something sometimes within an IRB?
Yeah, yeah, that's a good question. So, the task of the IRB is to establish the standard of care and to make sure that the patients, at least for the therapeutic components in the study, are not being deprived of the standard of care, or if they are being deprived of it, that they're getting something that's believed to be competitive. So that's that notion of clinical equipoise. So you're asking, do people always agree on what the standard of care is, or do they usually agree on whether the new treatment is competitive with standard of care? Are there debates about clinical equipoise? And the answer is yes. Oftentimes, for a lot of areas of research, it's not that controversial, but there are areas of research where it can be really controversial. There's a cancer drug called vemurafenib, a BRAF inhibitor. You guys are like engineers, so I don't know whether when I say BRAF inhibitors, this is like talking a different language. You probably know what BRAF inhibitors are. Anyway, this was a drug that was a highly targeted drug, and the standard of care at that time was a drug called dacarbazine, which is horrible. It's just like, it basically kills melanoma slightly more than it kills you. And so, in this randomized trial between this really targeted drug that, had a molecular target versus this, basically poison, some people thought it's an unethical trial because it's so clear that it's better to be in the vemurafenib group than the dacarbazine. So there are cases where there is a lot of, debate or contention. I would say for the most part there isn't a lot of debate usually, but there are exceptions. And the exceptions tend to be when people use placebos. A lot of times people will say, oh, it's unethical, it's inconsistent with standard of care. And FDA sends kind of mixed signals because FDA says, oftentimes presses researchers to use placebos where physicians wouldn't normally, placebo is the equivalent of like not giving anything for treatment. And there are a lot of areas of medicine, like depression, for example, or anxiety, where psychiatrists wouldn't just leave a patient untreated. And so that's one area where you can get big debates. Another area that's probably relevant for you, for this group, because you're probably talking about more engineering applications, is in devices. So these are typically implanted surgically. So what's your comparative group when you do a surgical control? It's going to be a sham control. So that means the patients get surgery without the addition of the device or whatever the surgical procedure is that's expected to be therapeutic. Well, that doesn't sound so good. You're cutting someone open, putting them on, they're getting general anesthesia, and they get, they're getting nothing at all. So those are often debated as well. But I would say that usually if you sit on IRBs, sadly, those to me are the important issues. They spend a lot of their time, too much of their time, talking about the informed consent document. Other questions? Okay, that's good. Should I move on to the next step? Okay, good. So now I want to try to get through this quickly because I'm going to run out of time. I want to just kind of make a couple points. I said there were sort of two different reasons why medical research is ethically fraught. The first had to do with making sure that patients were protected from undue risk. The second one has to do with the fact that we ingest evidence when the evidence that we produce from experimenting on non-human animals in humans is, in a sense, ingested by every downstream patient who takes the drugs, uses the devices, the vaccines that are developed from that knowledge. And that means that we have to really find ways to make sure that the information we're producing is accurate information. It's providing an accurate estimate of treatment effects. And there are a number of different cases or episodes that have shown that we don't always do a really good job at protecting downstream users by making sure that we have high integrity with the kind of research we do. And I'm going to go through two relatively recent examples. The first is an example of a treatment for metastatic breast cancer that used high-dose chemotherapy with autologous bone marrow transplant. Smart physicians, I think they were based actually out of Harvard, came up with a great idea. Take bone marrow from women with metastatic breast cancer, bank it, and then blitz them with lethal doses of chemotherapy, and then return back to them the bone marrow. Very, very aggressive treatment. There was a small phase 2 study that showed some promise there, and on the basis of that study, it was an uncontrolled study, there was no control group. On the basis of that, this became the standard of care in around 1990 in the United States. The United States spent about $3.4 billion delivering this treatment, 43,000 women exposed to this very aggressive treatment. It took about 10 years to run a randomized controlled trial testing this treatment against what had been the standard of care pre-1990. And when the study was finally done, by the way, not easy to run a study like this. If you're a breast cancer patient, why would you go into the 1989 standard of care if you have the option of getting the 1991 standard of care outside the clinical trial? So very difficult to recruit patients for this trial because the standard of care had shifted on the basis of low level of evidence. Well, when the trial was actually published in the New England Journal of Medicine, it turned out there was no survival advantage to getting high-dose chemotherapy, autologous bone marrow transplant, but there was a lot more toxicity. And that really made clear just how critical it is that we have high level of evidence before we allow standard of care to shift and evolve. So that was one big episode. The other big episode happened around 2004, and this was when Attorney General Eliot Spitzer in New York investigated GlaxoSmithKline for having suppressed information, safety information about their SSRI, their psychiatric drug, Paxil. It turned out that there was evidence in their trials that adolescents and children taking this drug were prone to increased suicidality during the early period of taking that drug, and litigation ultimately revealed that Glaxo had withheld this information from publication. Around the same time, you also had reports that Merck had done basically the same thing with its drug, Vioxx, a pain drug, with respect to cardiotoxicity. And this led to a series of changes that upped the amount of transparency that we demand when people do research on human beings. So there's a number of other sort of scandals that drove home just how important it is to make sure that the information that we collect in human subjects is valid, but also that it is accessible to decision makers who need that information. So what I'm going to talk about here is sort of the things that we ought to worry about before the trial's happening, during the planning and the reporting and uptake stage. So first I want to say something really quickly about clinical development. I don't know how, what the level is of this audience, but I do want to sort of make sure people know some of the terminology here. So when you develop a drug, there's typically 5 stages of development. The first stage is preclinical research, which would be studies in non-human animals. If you have, safety in non-human animals, you have some promise of potential efficacy, you would then launch what are called phase 1 studies that are safety and dosage. Phase 2 studies are your first glance or peek at whether a drug, device, or vaccine is effective. And phase 3 studies are the kinds of studies that are large, randomized controlled trials that really decisively nail down the efficacy of a treatment in some kind of clinical aspect of disease. At that point, if you have efficacy and safety, the Food and Drug Administration in the United States would approve the drug, and then there's what's called post-marketing research, or phase 4 clinical trials. So most of what I'm going to talk about is in that red space, the preclinical to phase 1 because I suspect in this audience, that's probably the most relevant area, unless you guys are all physicians, which I don't think you are. Probably mostly what you're doing is kind of preclinical research that's intended to inform phase 1 studies. And I want to start by saying that the vast majority of drugs that are put into phase 1 testing, this is also true of devices and vaccines, never make it through to FDA licensure. Only about 1 out of every 10 drugs that's put into human testing ultimately is shown to be safe and effective. In areas like cancer and neurological disease, the numbers are closer to 1 in 20 or 1.5 in 20, somewhere on that order. There are any number of examples of treatments. This is sort of a case in point in late 1990s, fetal mesencephalic tissue for the treatment of Parkinson's disease, where there was pretty good evidence in animal studies about efficacy. There was even really good mechanistic evidence and even good phase 1 evidence suggesting efficacy, but that when this treatment was actually put into randomized controlled trials using a sham comparator that was hotly debated at the time, it turned out that patients in the sham group had no difference in terms of efficacy, but had better outcomes in terms of safety. It turned out that patients receiving the fetal tissue had increased dyskinesia. These are these sort of writhing motions that Parkinson's patients have. So really important to rigorously evaluate studies, even if they look promising in preclinical and phase 1 studies, really important to put them through to proper randomized controlled trials later. Lots of cases where things fail in phase 3. So why do so many drugs that are put into clinical development ultimately fail? There are any number of different reasons, and I think some of the reasons are completely unavoidable. They're just a property of the nature of science. And the first point that I want to make about that is that whenever you do research on a novel drug, you're often doing research at the cutting edge of what you understand about the mechanism of disease. And so that means that oftentimes when a drug fails, it's because we don't really adequately understand the mechanism of disease. So Alzheimer's drugs, all of them have failed. They've been all motivated by the amyloid cascade hypothesis. They've all failed. And probably that failure reflects that we don't really understand exactly what's going on with Alzheimer's disease. So it's important to recognize that when drugs fail, because we don't understand mechanism, we're getting really important information from those trials about the mechanism. It's a chance to update our theories. So you're getting lots of useful information, even from failed drug development. The other point to emphasize is that a lot of failure happens because of the very nature of trying to search for effective drugs and treatments. So I always give this analogy. I don't know whether this will work among, you guys are all maybe a little bit young for this, but do you remember the Malaysian down jetliner in Indian Ocean? Okay. So imagine, you've got this down jetliner, and you need to find it in the Indian Ocean. That's a lot of geography to cover. So if you're going to try to find, a down jetliner, you're going to want to use a method that's going to scan the ocean very superficially over a huge amount of landmass, not invest too many resources in any one position in that area, until you find some flotsam or some debris, and then you bring the high-end sonar equipment out there to sort of look at the ocean floor to see whether you have something useful. So what does that mean for drug development? The analogy here is that think of the Indian Ocean as the equivalent of a vast landscape of combinatorials of drugs at different doses, in different patient communities, perhaps in combinations with other drugs. And think of that down jetliner as the one combinatorial that is actually useful. So if you want to scan that space to find something that's useful, not knowing that there's not that many useful products out there, you're going to want to use methods that are fast and frugal. What does that mean in terms of clinical research? It means you're going to use surrogate endpoints, so really quick readouts about whether a drug is effective. So in cancer, whether a tumor shrinks, as opposed to looking at survival, because it takes a long time to measure survival. And you're going to want to use frugal methods that aren't that costly. So you're going to use small sample sizes, small numbers of patients. Well, those are precisely the conditions under which you get false positives. But that's the nature of what scanning is about. If you have a scanning technology, just like looking at the Indian Ocean, you're going to want to very superficially cover a huge amount of space, and most of your positives are going to be false positives. So that's not failure because scientists are stupid. It's failure because we use methods to search a vast combinatorial landscape that are efficient. Nevertheless, there are a lot of practices in research that I think are that lead to failure that are very easily avoidable, I think, with some changes to the way we do research. And to sort of go through this, it's useful to think about the way we do research It's sort of a cyclical process where we design experiments, we report them, we interpret them and take them up, and then we design new experiments. And there are various threats to validity that get introduced in each one of these different steps. So with respect to design, every time you design an experiment, there are any number of different factors that can confound your measurement of the relationship between a treatment in animals or human beings and a generalized effect, right, an impact on disease. And so a key task is trying to control, mitigate, and eliminate these various sources of confound. And there are three different factors that you have to worry about when you design experiments. The first are internal validity threats. These are factors that might bias your measurement of the relationship of cause and effect. So for example, if you are unblinded when you do an experiment, you know which animals are getting the treatment, which ones are getting the control. Because you have an affection for your hypothesis, you want to get a paper into Cell, unwittingly, unknowingly, you score the animals in the treatment group a little bit better for outcomes, and you're a little bit meaner to those animals in the control group. You don't treat them as well. That's an easy way that bias can be introduced. There are any number of different practices that we use routinely in medicine to reduce those threats to validity that are rarely used at the bench. Randomized allocation. So how many people here work in a lab? Okay. So how many work with non-human animals? Anyone? Just one. Cells? Cells, couple? Okay. I mean, if you're doing a controlled experiment, certainly in non-human animals, but also to some extent with cells, oftentimes you want to use randomized techniques to determine which animals get treatment, which ones get control. You want to do a priori power calculations. You want to be blinded as to which group is getting treatment, which group is getting control. And these practices are relatively rarely implemented in laboratories. This was a study that came out a couple years back, but probably the numbers haven't changed that much. If you look at non-human animal studies in the medical literature, you'll see that about 0% of studies do a priori power calculations. Hopefully you guys are engineers and so you understand statistics. Most people in the life sciences really don't understand. I'm a bioethicist and I know more statistics than than most people at the bench. It's disturbing. I always ask on job interviews, what's a p-value? I think I've only had one or two candidates actually answer that question properly. Anyway, power calculations, I don't think even most life scientists even know what a power calculation is. Certainly it's reflected in the literature. People just say, oh yeah, I'll use 8 and 8 animals because that's what that guy did. That makes no sense. In terms of blinding or random allocation, about 7% of studies use randomized allocation with non-human animals. About 14% of studies use blinded allocation. So these techniques are not widely used in animal research. The second validity threat that we have to address are what are called construct to simulate a real-world setting. So if you do an animal experiment on a drug, you're trying to simulate what will happen when you give patients that drug. And construct validity refers to the concordance or the appropriate relationship between the scenario you're modeling in the lab and the one you're intending to model. So if you're testing a drug that's aimed at cardioprotection, and you lesion your animal, you cause a cardiac event in the animal, and you've started to give the drug before the cardiac event, and then you see a treatment effect, that is a construct invalid experiment because patients in the real world don't say, you know what, and I think in about 30 minutes I'm going to get a heart attack, I better start taking my drug, right? So it's important when we do experiments, if we're trying to translate into the clinic, that we model the animals in ways that are appropriate to the clinical scenario. Just for speed, I'm going to just skip through these next couple slides. The third issue are external validity threats. Essentially what this refers to is that when you see an effect in non-human animals, you want to make sure it's a robust effect. It doesn't reflect something idiosyncratic about your model system. Your strain of mice, maybe there's something quirky about their physiology, the particular treatment method that you used. So you want to replicate it under different varying conditions to make sure you have a robust effect. It's the same concept of robustness in statistics. You do a sensitivity analysis. It's the same kind of thing when you do an animal experiment. You want to kind of relax various assumptions and see whether or not you can get the same effect. And if you do, you have addressed threats to external validity. Your treatment effects are more likely to travel from non-human animals into human beings. And there's been a huge amount of literature over the last 10 or 15 years showing that lots of non-human animal experiments, when people go to the laboratory to try to reproduce them, fail replication. This was a study that looked at spinal cord injury treatments, 12 spinal cord injury treatments that had been, some of which had been introduced into clinical trials, and a laboratory funded by the NIH tried to reproduce those experiments. Only 2 out of the 12 ended up reproducing in the laboratory. So those are design principles. Reporting principles means that when you do the experiment, it's important to get that information out there. There are a lot of problems with the way we report research. One problem is we often don't describe our methods properly when we go to publish. Believe it or not, in my own studies, about 20 to 25% of preclinical experiments in animals don't even say what the sample size is. Hard to know how to interpret a paper if you don't have a sense of what the sample size is. That, again, speaks to the statistical illiteracy of many researchers. There's incomplete reporting. This means leaving out really key elements that are important for knowing what happened in an experiment. In a trial, for example, in human beings, typically you would report the number of patients randomized, the number of patients who have been observed, and the number of patients that have been included in the analysis. Almost every clinical trial you read will go through, have numbers in each one of those steps. I think you'd be hard-pressed to find any examples of that for non-human animal studies, even though those numbers might change, right? You might treat, 8 animals and 8 animals and accidentally step on one of the mice because you dropped it, and so 8 goes down to 7 in the treatment group, so you've only observed 7. And then one of them has this weird, anomalous reading, so you don't include it in the analysis. So 7's gone down to 6. If you have a small sample size, 8 and 8 animals, your number, you have very limited statistical power. Your analysis can change dramatically because you've left 2 animals out and you haven't imputed the missing data. The last point, and this is probably the biggest problem, is just that a lot of research is just simply not published. It stays in laboratory notebooks. There are various statistical techniques that you can use to measure the amount of research that's not published, animal research. These methods are probably conservative. There have been various studies that show that probably if you had published all the literature out there, for lots of treatments, you would have reductions in effect size of at least a 30% or 40% reduction in effect size. And I think those numbers are really quite conservative. So that's a big problem, is that people don't publish negative results, but they should be. And then there's the issue about uptake. This is really disturbing, but it's true. People don't read. It's amazing. We learned this in first grade. And somehow a surprising number of people who are academics lose their ability to read. And so there's a lot of actually informative literature out there that actually could be useful if people actually took the time to do proper PubMed searches, read the articles carefully, and incorporate it into their thinking. This is just one small example. This is a disease called inclusion body myositis. This researcher was really peeved at the way that people portrayed the pathophysiology of this disease. And so what he did is he did a citation analysis where he took every claim that had been made in the medical literature about the cause of this disease, and he wanted to see what articles were being cited and how those claims were cited in subsequent articles. So he did a network citation analysis. And this just looks like a total mess. This is what he found. Essentially, there are 5 studies that support one hypothesis. There are 6 that disprove the hypothesis. Almost all the citations are to the ones that support the hypothesis, relatively few citations to articles that disconfirm, even though methodologically the disconfirmatory studies were indistinguishable from the confirmatory studies. And he also found what to me is really amusing, and if you've been in academia, you know this has happened because you've probably had it happen to yourself, what he calls citation transmutation. This is when a study that is claiming X is cited as if it were disclaiming X. So, I've had this happen where I've argued this is unethical, and then I'm cited as saying this is ethical. So that's citation transmutation, which happens a surprising amount. Okay, good. So let me turn now to talk briefly about why this matters. Okay, I mentioned human beings have moral status. They suffer when you do experiments on them. It's the same thing. Same thing is true for non-human animals. I mentioned this for patients already. This is something we don't talk about enough. You guys are probably here on NIH money, I assume. NSF? Right. So the taxpayers have paid you have paid me to come out here to train you. So there's been a huge amount of accrued social investment in your training. And if your effort is allocated towards unproductive research avenues, unproductive in part because we're not using techniques that are allowing us to make clinical generalizations, we're not getting good return on that social investment. And you guys are a scarce resource. It might not feel like it to you, but you are. There are not very many people who have your expertise. And so as a taxpayer, as an ethicist, I want to make sure that your talent is applied in ways that are going to maximize productivity. So that's another reason why we worry a lot about methods that are not reproducible methods in laboratory research. And the last point I want to make is that it's tempting to think that the work that you do in the lab doesn't really have that much relevance for patients. I mean, if you're in grad school, you probably think you have no relevance to patients. But I have good and bad news for you, which is that actually a surprising amount of bedside decision-making is made not on the basis of clinical trials, but on the basis of animal evidence. You know, we live with this fiction that, that diagram I showed you before, that we do the preclinical studies and you have to do phase 1, phase 2, and phase 3, and nothing gets to the bedside unless it's gone through that gauntlet of really rigorous testing. And that's just simply not true. There are a lot of areas where there's a lot of bedside decision-making made on the basis of laboratory experiments, non-human animal experiments. I'll just give you a quick example. This is a drug called bexarotene. This was a drug that's approved for a rare kind of lymphoma. And there was a study in Science magazine that's reported that this drug could actually reverse some of the biochemical deficits in Alzheimer's disease. Great publication, 2012. On that basis, lots of neurologists were beginning to talk about the prospect of using this drug off-label. In fact, New England Journal of Medicine ran an article about the ethics of using this drug off-label. That was only on preclinical evidence. Because the drug was already approved, doctors are free to use the drug however they want. There was no trial evidence at all. There's an interesting footnote to this story, which is that it turned out that there were lots of labs that had a hard time replicating that Science paper. And so I don't think anyone really thinks that bexarotene actually works in Alzheimer's disease. So kind of a good example of the ways that reproducibility threats can ultimately end up with patients taking ineffective drugs. We literally ingest the information. Okay, so what can you do about this? I want to stop after this segment here. The first thing I want to say is be scared. The bad news is that you can't say, you know what, FDA's got this, they've got my back, nothing I can do, it's going to harm people, because the FDA, we've got good, competent regulators that are making sure that patients are protected. FDA is actually quite explicit that they don't really care that much about the evidence that a drug works before you start testing in patients. They care a lot about safety. So, they'll have your back for toxicity, but if you say, okay, this device, has promise for, myocardial infarction or, Parkinson's disease, a particular cannula, whatever it is, FDA is like, don't worry about the evidence, of efficacy. I just want to see the safety. And in fact, this is a quote from a very, very prominent person at FDA. I mean, Bob Temple. If you follow FDA like I do, he's sort of like basically runs the division on drugs. He says very explicitly, this was at a meeting I attended in 2016, that it's up to sponsors to decide whether or not a drug or device is effective enough to put into animals. So are there examples of treatments that are demonstrably ineffective that have been tested in human beings? Yes. So here's an example of a study that was published for a cell therapy for treating spinal cord injury. The trial was a phase 1 trial. It was stopped because the drug or the cell therapy didn't seem to be very effective. And after it was discontinued, the researchers who had done the preclinical experiment published a paper that showed that the particular cells that were used in this study were not effective for controlling spinal cord injury. This information was available to the company and to the FDA before the study started launching, and yet the study went forward anyway. There are other examples of this out there in the literature. So the authors of this paper had the conclusion these data raise questions about the lack of FDA requirements for in vivo testing of intended cell lines. Okay, so what can we do? How can we improve the design? Well, there are a number of guidelines that are out there. For example, in different disease areas, there's one in stroke, but there are others that are out there about how you ought to design preclinical studies if you're intending to support development of a product into phase 1 or phase 2. If you want to read it, I published a meta-analysis or a systematic review in PLOS Medicine. I think it's 2013. It's got truncated a little bit. That reviews these various techniques you can use, randomization, blinding. When you publish studies on non-human animals, there are guidelines out there about how you ought to report them so that people can adequately interpret them. The ARRIVE guidelines are probably the most famous ones. They're endorsed by well over 300 medical journals. Nature, for example, if you submit to Nature, basically you have to comply with ARRIVE, Cell as well. I realize these are probably slightly different journals than you guys are going for, but you've heard of Nature at least, I'm sure. Also, you can prospectively register your studies in, there's a place called Center for Open Science. Anyone ever heard of Center for Open Science here? Okay. This is a great resource where you can take your protocols and register them before you do the experiments so that you can't go back and p-hack. You can't go back and pick through your data to find a positive story in all your data collection. And so this is a great way to protect the integrity of your research. In terms of uptake, what can we do to improve uptake? Try doing systematic review. There are a lot of resources out there about how to do systematic reviews. You know, I'd say that if you're doing any kind of research that's intended for human application, try doing a PubMed search. And try to debias yourself by looking for evidence that disconfirms what you think. Look, specifically target trying to find information that's going to disconfirm your hypothesis. Okay, so I'm just going to close with just these quick remarks. What's the take-home message of all this? This is the second part. Most treatments that are put into development fail development. Some of the failure is statistical. It has to do with the nature of the scientific process, but some of it is avoidable, and it has to do with poor practices we use at the laboratory bench. Every time drugs fail, it means that there's been scarce resources wasted, and it means the patients have been exposed to a potentially ineffective and or unsafe intervention. And I've described a couple things that you can do to try to reduce some of that burden. So the last point I'm going to leave is that none of you guys are ethicists, and it's tempting to sort of have a view on the world of ethics that looks something like this. You've got the technical decisions you guys are dealing with, how you set up your experiments, how you power them, what your sample sizes are. And then we've got these, sort of this ethics overlay, informed consent and, is there good risk benefit, et cetera. And I want to encourage you to think of a world where the ethics and the technical are fully integrated, together. That every technical decision you make, I know it's hard to believe this, is saturated with ethical judgments. So the example I always like to give, so again, I assume you guys have some statistics, is if you ever do a power calculation, do people know what I mean when I say power calculations? Okay, I see a couple heads that are bobbing, that's good. So, you're picking an alpha and you're picking a beta. What you're doing when you pick an alpha and beta, that's not a technical judgment. You're deciding how much am I willing to tolerate the risk of false type 1 error or false positive. How much am I willing to take a risk of false negative. Those are value judgments that embody judgments about ethics. And so right there in the architecture of your study and how you're doing it, you've made a series of ethical judgments without even knowing it. And so I think it's really valuable to understand that almost every design aspect of any experiment you do is embodying some kind of ethical judgments. Thanks. I'll stop there and take questions.